Hi, in this work, we explore the role of cis-regulatory motif syntax in human somatic cell reprogramming. Somatic cells, such as fibroblasts, can be reprogrammed into embryonic stem cell-like cells by inducing the four Yamanaka factors, OCT4, SOX2, KLF4, and MYC. We begin by profiling cells with single-cell RNA-seq and single-cell attack-seq for approximately 50,000 cells each, sampled every 2 days across a 14-day time point of fibroblasts induced with the four Yamanaka factors. In addition to a primary reprogramming trajectory, we observe multiple non-reprogramming trajectories. Interestingly, the initial heterogeneity can be explained by differences in the levels of the four factors expressed in each cell. We then train base resolution neural networks that take in a 2 kilobase DNA sequence input and predict the attack-seq profile of TN5 transpose on cut sites in a cell state-specific fashion. We train one model for each of our cell states. We can then interpret these neural networks at base resolution and obtain cell state-specific motif syntax maps at any enhancer of our choice, which highlights the subsequences that are most important for the attack-seq signal in that cell type. By synthesizing these motif syntax maps across modules of cis-regulatory elements, we observed two interesting phenomena. We observed a new class of motifs that we term anti-accessibility motifs, whose presence is predictive of lower attack-seq signal than their absence. These motifs include the ZEB and the ZBTB7 factors, both of which are known transcriptional repressors. Finally, we also observe that transient cis-regulatory elements that are exclusively accessible in the early phases of reprogramming contain low affinity OX, SOX, and KLF motifs that are bound only at high expression levels of OX, SOX, and KLF. If this looks interesting, or if you want to discuss reprogramming, motif syntax, or neural networks more broadly, please reach out to me.